Okay, so today what I will try to do is I will try to convince you that imaging single molecules is has some, if not a lot, but at least some advantages over other regular quantitative methods. If you think about imaging single molecules, you are really doing ultimate detection. Right? There's nothing smaller than there's no half molecule. So we will see what are the requirements. What do you really need to do to be able to see single molecule? What do you really need? I'll discuss some of the techniques that are commonly used, though sometimes briefly because some of them have been covered to some certain extent, but feel free to stop me if there is something that hasn't been covered before. And then what I will do is focus instead of just the optics, but also on the how single molecule methods or imaging can be used to interpret the data, or what properties can you use of the single molecules to infer some biological phenomena. Okay? And I will, at the end, try to show you two case studies where either single particle tracking or super resolution imaging has been used in a certain way to infer. And today we will focus on quantitative interpretation of what is might be going on. Okay, so see what methods allow us to get quantitative with some of these techniques. Okay, not just pretty pictures, but really get something really quantitative, which can be used for mathematical modeling as well. Okay. So the reason I talk about this picture when we are talking about single molecule is because I want to give you a glimpse of what you're really trying to do. So imagine that this is a biological cell, and you have several proteins, some in different numbers. Each of the proteins have their individual jobs and if you take, if you look at the function of the cell, what you're doing is you're just listening to the music from this orchestra. If, however, you are able to take a snapshot from a very high up position in this hall, you will see that there are things going on, things moving around, things being done at different times, but interpretation is still harder. Till you get to a stage where you can see these individual performers, or in your case, individual proteins, that you understand that what those might be, and you usually try to infer them in imaging by looking at where they are localized, where they are, when they are performing, and what they might be performing. Right? And their problem is that not only there is heterogeneity in different kinds of proteins, even in single proteins, there's a lot of heterogeneity. And the consequence of that is, and they're kind of small to start with. So this is a snapshot of a small region in, from a bacteria. It's a schematic, of course, drawn to scale. And you can see it's highly packed system of molecules, each interacting with other, and of course has a lot of intermixing and interplay between all of those molecules. And each of these molecules themselves can display different properties at different times, or within a population itself, because of a post-translational modification, their kinetic properties can be different. So here's an example of two molecules which are exactly the same otherwise chemically, but for some reason are showing different kinetics, even if their equilibriums are exactly the same. So this is the actual data taken from a single molecule experiment. Okay, don't worry about what those traces mean at this moment. I'll come back to them later. So this is what we call static heterogeneity. And of course, any process, any biological process and chemical process that you take, and you try to initiate the reaction. So in this case, there's a protein unfolding study going on. When you initiate, so you start with all folded proteins, you try to initiate unfolding. Each of these proteins will be at a different stage of unfolding and will eventually reach a completely unfolded state. But when you take a snapshot at any time point, you will see a mixture of those. And then you have to be very carefully interpret the data in terms of where there might be some intermediates, where there is slightly longer lived intermediates that you can use. Okay. This is not required, at least this kind of a complication doesn't occur if you did have, so if I just took, oh, this is, if I just took one of these proteins and just looked at their unfolding, then they will go from a state A, let's say, with high signal here, with, I'll come back to again what these signals are, and then they will see intermediate that might be short-lived eventually, and then things unfold, right? If you had taken this ensemble data, then what you would have got is this very smooth curve where you would have to fit models, come up with different models and fit this data and try to infer if there is indeed an intermediate in this unfolding pathway. And people have become very, very good at interpreting these things by using several methods. But if you do a single molecule experiment, you don't need to fit a model, you will see it directly. Okay. Okay. And the other thing that people have found with single molecules as well is that most of these proteins and DNA molecules themselves are big polymers, and they are folded in a three-dimensional state, but they are undergoing fluctuations. They are undergoing fluctuations because of the thermal path they are suspended in, and that causes them to change their configurations just by a slight amount, and that's sufficient to change the activity of that protein. So here's the activity of a single protein that has been measured over time, and so this is beta-galactosidase, and these fluorescent spikes that you see are the activity of the protein, and you will notice that this protein has time windows over which it is highly, it's a very efficient enzyme, and other times it is not doing much at all. The reason for that is it is fluctuating between different configurations, and the active site is not stable enough, sufficiently stable. And this is roughly the story for almost every molecule that you encounter. Okay, and this can have different implications depending on the questions you might be asking. Okay, so there's one more reason to aspire to have sensitivity of imaging single molecules. If you look at many proteins inside any cell, whether you take bacteria for that matter, whether you take yeast for that matter, and you measure, of course, these are measurements either most of the time done in ensemble, but sometimes at single molecule level or single cell level, and you try to estimate how many copies of molecules you have, you will find that the average number of proteins, for example, in bacteria, is about close to 10 proteins, okay, of any particular kind. Okay, so many of these proteins are not being studied because you don't have methods to study them. If you overexpress them, then it's not the natural phenotype. So most of the time, these are not being studied. If you had a way to image them at single molecule level, then you would be able to do this experiment. So that's why half of the biology is still hidden, because you don't use single molecule methods. And this is roughly true for other systems as well. So transcription factors, this is log2, by the way, are present in a few tens of hundreds of copies inside the yeast cell as well. And depending on several of the other biological systems as well, these numbers are extremely small. And the consequence of this is that when single molecules are present inside the cell, or they are so low in number that if you have a small fluctuation in the number of molecules, you can go from a state which is a particular kind of a, let's say, a zero state where the molecules were very small in number, but because of some feedbacks that exist in the system, these numbers can very quickly fluctuate, turn on the feedback, and then go to a completely different state. And this has been experimentally shown for isogenic bacteria, where if you have, let's say, the lac operon, you can get in intermediate concentrations of the inducer, you can have bacteria which will be producing the protein, but will not switch on and can get induced all the way. Okay, so you have a completely bimodal population just because of fluctuations in single molecules. Okay, so isogenic bacteria are two different populations coming just from the fact that single molecules are fluctuating between these systems. Okay, and same goes for mammalian systems as well. What happens is transcription factors would bind for a very short period of time. You will produce mRNA and then the proteins will be produced, but only till the transcription factor was bound. And then there's no transcription factor bound to the mRNA, and the protein, of course, has their own decay times. They will decay back till the next transcription factor binding event happens, and then this will fluctuate. So instead of imagining that there is a steady state of many, many proteins, what you see when you try to measure gene expression, protein production from a particular, or protein levels for many of the proteins, you'll see them fluctuate between high and low levels. And this is more realistic than imagining a steady state for which happens, but only for a very small fraction of proteins. Okay, so we are only asking these questions now because we are getting sensitive methods to image them. Okay, now what I want to try to convince you is that those fluctuations are coming from single molecules, and so you need to be able to study those single molecules which are defining the or determining the decision process. Okay, and to be able to image these single molecules, you need, according to me, at least 4 things. All right, these 4 things are listed here. You need well-behaved, and I'll come back to the details of what I mean by well-behaved fluorophores. You need background suppression. I will again explain why background suppression is very, very important and how do you do it. You need high quality optics, and when I mean high quality optics, it's not only transmission of the system, but also numerical aperture of the system, which allows you to collect the number of photons, the largest number of photons as possibly as large as possible from the same single molecules. And you need sensitive detectors, which are most of the time have to be sensitive to single photons. And then there's a trick that we use quite often. I will explain this in detail as we go along, which is what I call distribute and immobilize. Okay. Why is this needed is something that will become clear to you, or maybe some of you already know what I'm talking about. Okay. I'll go through each of them and then I'll discuss in detail. So fluorophores, most of the time you don't want to take a simple dye that you have been using quite often. The reason you have been using it is because it's a very easily available dye. It's easy to conjugate, it is cheap. And if you use that dye, you might or might not see single molecules. It will depend on which dye you pick. Many dyes will not give you single molecules because they either do not absorb sufficient light that is thrown at them, or the quantum yield is small such that they don't convert the absorbed light into sufficient number of emitted photons. Okay? The second thing is those dyes might not be photostable, and if they are not very photostable, then they are going to kind of disappear even before you can collect sufficient photons from them in your microscope. Okay, they should also show minimal photophysical effects and aggregation effects, and this is something that I will come to. Usually, good single molecule dyes would have about what we call a photon budget of about 1 million photons. 1 million photons released before they kind of disappear. If they do that, then they are a good quality single molecule dye. Okay, of course you don't need to measure this. You will find in literature which dyes are more commonly used, and I'll discuss about some of those. The other thing that you want is, of course, if you're introducing it into a biological system, most likely a protein, it is a cell that is something that you're trying to label. You want them to be small so that they don't perturb the protein or the DNA that you're trying to tag it to. You want them to be water soluble and want sufficient forms of conjugating it to the molecule of your interest. So here's a list of some of the favorite dyes that people use around the world. They're the cyanine-based dyes. You have heard about many of these talked about. They are these rhodamine-based dyes, again, quite popular. This is Genelia farm dyes that came out. a few years ago. And then you have also heard about fluorescent proteins. Of course, there are GFP-like proteins that are used. So this is one of the brightest ones that I've come across recently. Then photo switchable or photoactivatable fluorescent proteins are also quite bright. Many of them are quite bright. And then the redder versions of these dyes, fluorescent proteins, are also quite commonly used in small molecules. Okay. Quantum dots, almost all of them, if you are interested in just tracking molecules or larger objects, not proteins. I don't recommend them for pure proteins, but motor proteins are commonly labeled tagged with quantum dots, and they are studied by tracking them in live cells quite often. If you have an opportunity, there are ways of suppressing the blinking that is very common in many quantum dots. If it doesn't matter for you, then let's go ahead and use it directly. But if there are ways to suppress blinking, like using high-all groups attached to quantum dots, or having a solution which contains like a PME or something similar. Okay, and there are more exotic molecules like the nitrogen vacancies in diamond clusters, and if you are looking at scattering, you can also use gold clusters, and they seem to also be robust tags for single molecule. They will give you sufficient scattering of fluorescence photons that you can observe them at single molecular. Okay. So while you talk about these fluorophores, you need to keep some of these points which have been discussed previously in some way or the other, and what limits the usage of these fluorophores. So for example, if you are interested in knowing how fast can I do my imaging, okay? So how fast can I do this? What's the time resolution that I can get in a single molecule? Remember that each of these molecules are getting excited to the excited state, and then they do interternal conversion, get to the first excited state. From there, they are coming down to the ground state and emitting photons. But that's not the only thing that they do. Okay, this lifetime, this is the time that it takes to get there and then to come back, is of the order of a few nanoseconds. If you have a lifetime which is very, very long, as the dye is going to take as longer to do the same cycle, and that is going to limit your time resolution, number one. If that dye is going to triplet states, which are usually dark states, they are going to reside in these states for sometimes as long as tens of microseconds, hundreds of microseconds. Those are times that are lost while you are trying to image that molecule, and that will add to a noise in the emission from that single dye. And now you don't want the photophysics of the dye to be so variable, not variable, but variable, that you can't infer what changes are happening in the system which you're trying to study. So you want to avoid dark state dyes that go to dark state for long periods of time until there is a specific reason for why you want a system like that. If you keep hitting those molecules, you will get to a stage where you are competing with photobleaching, as well as you will be reaching saturation, which is the number of times it is cycling through these excited state and ground state. Those will reach a stage where you won't be able to pump in more laser and get it to go through this cycle more number of times, because they're already running at maximum duty cycle. So those things also limit your ability to do this. And then you don't want to pick up dyes which will give you different kinds of excited states and emission state, because that makes your interpretation of this data harder. Many ways exist to increase the photostability of the dye, and then those are things that you can take advantage of. So many oxygen, for example, produces radicals which are highly reactive, and these, if you remove them from your system, from your solution system, either by degassing or everything into a nitrogen environment, you will make your dyes longer than usual. And there are enzymatic systems which are much more commonly used because of the fact that they are much easier to implement. So you just make a cocktail of enzymes and add it to your solutions if you are doing an in vitro experiment. And sometimes people have been able to now add some of these molecules directly to the dye, like Scott Blanchard's lab does, and have been able to stabilize dyes. And I want to show you the effect of these dyes and the photostabilizing systems. So if you, for example, notice the data that is kind of shown here, so in seconds, you are able to, for example, double your time for which you can observe a dye, more or less sometimes as much as 3 times, just by adding the right oxygen scavenging systems and triplet state quencher. Okay, so the triplet state, this is a triplet state quencher that is doing the same job that normally an oxygen does, but because you have taken away the oxygen that produces free radicals, now this dye is able to do give you consistent level of photons for a much longer period of time before it photopleaches. So tricks like this can be used to stabilize the dye from photopleaching, and this is important because you want to increase the amount of information you can get from a single molecule. You can also have to think about linking the dye, and there are 2 types of systems that we have studied. Of course, there are these organic dyes that can be linked by covalently attaching them. The standard chemistries are either using an amine reactive group like a lysine is tagged, or a thiol reactive group like cysteine is used by some of these chemistries. Or you have heard about genetic tagging, where the protein backbone is either modified using an unnatural amino acid. You can also create a fusion protein with a taggable protein like a snap tag or a halo tag, and then of course fluorescent proteins. If your proteins are available in, on the surface of the cell or easily accessible, then you can of course tag them with nanobodies or labeled antibodies, or the tag could be in the form of a DNA paint system, which you might have also heard about. Now, which chemistry do you use? That depends on the protein and the system that you're trying to tag. And where do you place a dye also depends on the question you ask. For example, if you're trying to do single molecule FRET, where the exact location of the dye matters because it's such a short distance process phenomenon, then you might have to be very careful about where you place your dye, otherwise you will never see energy transfer. But if you're just trying to tag it to a place where it doesn't interfere with the function, but you only want to be able to track that protein in real time in the milieu of the cell, then it doesn't really matter where you tag it as long as you don't lose function. So these are project-specific questions, and I'm happy to answer them if you are interested at a later point. Once you have got your dye and you have figured out how to keep them alive, you still need to do what I call background suppression. The background suppression is most of the time when you're trying to do this imaging, you have other molecules which are fluorescent. If you are doing it inside the single cell, there are autofluorescent molecules present, which might not be as bright as your single molecule, but they are much larger in numbers. So they contribute, they add up, and then they give you background. And that kind of kills the contrast that you will have for your single molecule. So you want to able to reduce that background. And so epifluorescence is usually the least preferred way until your cell is really a dark cell to start with. Most of the time, this is never the preferred way. The more common ways that people do imaging is either doing this highly inclined thin illumination, the high-low method, or if your protein or your system is close to the cell membrane, then you would use total internal reflection fluorescence microscopy. Okay, zero mode waveguides are used, but they are most commonly used for in vitro systems where you have purified systems and you can get very good suppression of your. I'm not going to go into details of zero mode waveguides or neither epifluorescence because you have seen a lot of them. All of you probably have epifluorescence microscope in your lab. Confocal microscopy is used, but confocal microscopy, as you have probably understood by now, suffers from time resolution. So it's great for taking images, pretty images, but it is not great for taking fast, high resolution, high time resolution data, which is something that you might be interested in because the molecule itself is kind of moving around, doing some dynamics, which is what you're trying to capture. So most of the time they are wide field methods, either by using high-low illumination or the total internal reflection fluorescence microscopy. So those are the common techniques. And they are really cousins of each other. You've probably heard this before, but I just want to re-emphasize this point. So epifluorescence, what you're trying to do is on your objective, you are focusing at the back focal plane of your objective such that the beam that comes out is kind of collimated, and this dimension is defined by the objective here and the lens that was outside, which is usually this lens out here, which is focusing things onto your back focal plane. Okay, so it's acting like a telescope of some kind, this objective and this lens out here. Okay, in TERF, you have probably heard before, but I just want to re-emphasize that you take this beam and instead of sending it right to the center of the objective, you move it to a side all the way till it is hitting this interface, which is the interface between the objective and the low refractive index media. So if it is water here, then you need to have oil out here. Okay, so that's where it is going to do the total internal reflection. So this is the objective type TERF. There are other ways of creating a TERF, but in that case, the TERF evanescent wave will be created on a completely different interface, not on this interface. So this is what is called the objective type TERF. You're hitting this, most of the light is reflected back, and only a small evanescent field travels out here. Usually, this is of the order of 10 to 20% of the light that was sent across. So this is why when you're doing TERF, you need a high-end or a high-powered laser. High-low is, in terms of its position of this beam, is somewhere in between the epi and the TERF. What is happening is, instead of doing this very sharp angle, which is larger than the critical angle, you have just moved it from the center by amount such that this beam is coming off at a highly inclined angle. And you can also reduce the amount of Na that is being thrown at this by cutting down the beam that is coming in. If you do that, the part which is illuminated can be made extremely small. So what is happening is a small pencil of beam is coming out at a very inclined angle. And if there is a cell sitting out here, then that beam is literally sectioning it, though it sections it at a small angle, but it sections it such that nothing else is illuminated above the cell, or nothing else is illuminated which is in the near periphery of the cell. And this is also commonly used to get high contrast, especially used in the single molecule and super resolution fields. So can I do a microscopy of single molecules on my current microscope? That will depend on some of these answers to some of these. Do you have a laser in this range? Is it illuminating your microscope in this high-low version or the TERF version? So you need to be able to reach this kind of power densities to be able to extract sufficient photons from these single molecules. So if you don't have that, then you won't be able to get sufficient photons in the time that you require before it photophages. Your objectives need to be high in Na, high enough in Na. Something in this range is what is required, and they need to be high in transmission range, which most of these objectives are in the visible range, where your fluorescence will be. Your filters and your lenses need to be achromats, and they need to again have high transmission in a visible regime, which is usually true. But about the filters, you can go and look them up. There are filters and there are filters. So you want to have the highest transmission for your visible range. You will need to spend some money to get them, but this is what you are aiming for. So this is about 95% or higher transmission in your range for your dichroic as well as your bandpass filters, right? There's the excitation filter, there's the emission filter. But not only that, you need to have these things block out. So you want ODs higher than 6 to block out all your laser intensities or any other stray light that will be coming in as well. This is important because, as I said before, you are working really hard to collect all the photons, and any photons that is not your single molecule's photon is going to act as a background, and that's something that you want to avoid. So that's very, very important that you get good filters for your microscope. Bodies are not most of the time important unless you have a lot of area that you need to sample. In that case, you might want to have an automated stage, but microscope bodies, otherwise, you really don't see those single molecules. You do see a cell, but you don't see the single molecule. You can't focus on it. You have to rely on fluorescence imaging for doing that. And your detectors, as I said before, have to be extremely sensitive. Some of the common detectors that are used by the single molecule community is kind of listed here. Of course, they are representative, and I have nothing to do with these companies. But these are most commonly used. You will find newer versions of these as well, which are coming out from day to day. You can, in confocal detection, use APDs. They are better than regular PMDs, and they will give you a single molecule. They have single photon sensitivity, so they will give you a single molecule detection. Okay, so confocal based single molecule is also commonly used, but the tricks are slightly different there. And if somebody is interested, I'm happy to talk to them about it. But we'll talk about where we are creating images of single molecules. The other trick, this is the last trick that you need to have, is that you can't see single molecules together. You can't see them if all of them were together in one place. And the reason for that is, by now you already know, is that the diffraction limit is already, even if you have a 1 nanometer molecule, you are going to see an image of that, which is going to be around 200 nanometers wide in a microscope or your detector, right? No matter what you do. So the only way to get around it is by doing STED imaging. But it usually kills the molecule before you can take a nice, great image of it. So there are very few single molecule STED papers out there. Now, so if you want to study a molecule and you have no problem in diluting it because it's an in vitro system, then you dilute it, keep diluting it, till you reach a concentration where each of your single molecules can be well separated from the other. Basically, their average distances have to be much larger than the 200 nanometer diffraction limit that we are talking about. The other trick that people use is selective activation. I'll talk about them in a little bit. And you want to be able to immobilize these molecules because you want to look at them for a very long period of time. And if you want to do that, then yes, you don't want them to be diffusing away from your imaging volume. So many single molecule spectroscopies and imaging folks rely on this property that the molecule either has been covalently or by biotin-avidin interaction attached so that you can follow this molecule, collect photons from molecules for as long as possible. Or they take advantage of the fact that a molecule which is free in solution, once it binds to this, will diffuse much, much more slowly because it's literally going to be immobilized or moving along this molecule if necessary. And this way, you might be able to get photons from it when it's interacting with the molecule of your interest. So that's another trick that is commonly used. If you don't want them to be attached physically, people also entrap them inside nanocontainers, vesicles that allow them to be free inside a volume, but still immobilized in a diffraction limited spot because these vesicles themselves are no longer or no larger than 100, 200 nanometers. Okay, so tricks like this allow you to do the imaging. And if you're wondering how are these, where are these things, So again, they are immobilized on the surface of inside a microfluidic channel that has been created using cover slip and a double sticky tape if you don't have access to photolithography for making these kinds of systems. So you introduce your molecules from this end, they will immobilize because some of these systems have been prepared beforehand chemically, and this kind of a chemistry allows you to immobilize the molecules. You add only so much concentration of proteins such that, or DNA or a lipid, whichever way you're trying to image, such that you only get single molecules partially separated from each other. Okay. Okay. So now I talked about what happens to a situation where you have a lot of molecules and there's no way you can dilute it any further, let's say inside a cell. And there we, of course, get helped by our ability to selectively activate a certain number of molecules. So while trying to do single molecule experiments and or trying to develop proteins which will allow us to timestamp or activate or pulse phase fluorescently systems, people came up with methods where you can convert a dark molecule to a bright molecule by an illumination, or a bright molecule to the dark molecule with another illumination, or a small molecule like small chemical like let's say mercaptophenol. So this is a paper from Chao Zhong's lab. So this is the cyanine dye. It's emitting here. You can see the single molecules. You illuminate it for a small period of time using BME or cysteine. They will go to the dark state. However, they do have absorbance in this regime. So if you hit them with a 405 nanometer or a 350 nanometer light, they will absorb it very, very quickly and then switch back. So you have now a way to illuminate some of these molecules. The trick is you don't illuminate all of these molecules. You switch on their illumination to at very, very low intensities that only a subset of these molecules switch on. So even if you have hundreds of molecules, just like in super resolution microscopy, you want to switch on just a few of them. If you do switch on a few of them, now you have that single molecule to interrogate. You let them photo bleach, and then the next set of molecules can be interrogated. And this trick has been used in the single molecule biology or biophysics field quite often. And the similar idea can be used with the photoactivatable system. So here is what your data is going to look like. You have activated your molecule, but only a single molecule in this whole cell has been activated. It's not as if there are no more drone for molecules inside the cell. They're also fused to some molecule of your interest. And then you can track it for some period of time. That molecule photo bleaches. You switch on the laser again, but this time again, the intensity is low enough that only another molecule switches. Of course, you don't have a control over it. So your intensity is kind of a representative intensity that allows only a single molecule to switch on, right? And then you track these molecules. And this kind of techniques have been kind of expanded where you can tag them with other dyes, which will allow you to give laser intensity to, let's say, this molecule, then activate any of the other molecules, and each of them can be addressed using the same laser, but different colors, emissions can be detected. And you have a very large range of molecules now in the repertoire of chemists that allows you to do this both for fluorescent proteins and for organic dyes, that allows you to switch between the on and off state. So the trick is you do limited activation, do not switch on all the molecules, and then acquire data. Okay. So coming back to why this is important. So here's an image of your single molecules, let's say immobilized on the surface, and I have been able to dilute them out. If I was not able to dilute them out. Can I just interrupt? Yeah, Devalina, go ahead.
So like in the previous section, when you were talking about activation pulse, yes. So there we saw the mechanism that molecule 1 gets activated first, and then it gets photo bleached, and then molecule 2. So how this track is being maintained? I mean, what if I excite the similar place multiple times? I mean, how to
good
distinguish?
question. Okay, good question. So you can't. In this case, this is why it is also in the super resolution technique that you know about. It's called stochastic optical illumination. What is happening is most of the time, so this is, I have drawn a bacteria. In fact, this is from a paper by EU, where a bacteria, and you know that bacteria itself is about 2 microns wide, 1 micron. So there are very few ways that you can come up and illuminate only this particular molecule. What you instead did was your beam was sitting on top of this region somewhere here. Your intensity of the laser was small enough that you got only one molecule to switch on. Okay, I don't even know, there might have been another molecule in the neighborhood vicinity. We could not switch it on, we switched on. But once we have switched that molecule, all the properties of that molecule can be measured. I will come and discuss what are the properties that we can measure for that single molecule. That photo bleaches, I have no control over when it photo bleaches out. Okay, and I have no control over when the second molecule is. So that molecule is still there, by the way. That protein, even though it's not emitting light, it's there, just that you can't see it anymore further. The second molecule that has been switched on, again, I have no control over which one got switched on, because again, the bacteria is too small. I can't control the light to that particular molecule. So I have no control. That is happening stochastically. Did I answer the question?
Yes, I got it. So basically, things get controlled during the data analysis part. I mean,
yes. So you infer things at the data analysis part, and you cannot infer which molecule is switched on and which molecule you switched off. That is happening randomly. Okay, but there's still a lot of things that you can learn, and that's what we will talk about today.
Okay, thank you.
Okay, all right. Yeah. So as in fact, this diffraction limit is one of the reasons why we cannot. So these two molecules are about a micron away. Their images are about 200 nanometers in sizes, roughly. I know that for a fact because each of these pixels is about 100 nanometers in my microscope. Okay. So this is how I will see them. But if there were two molecules which were closer than this distance, as you would imagine, even if the molecules themselves were not even touching each other, they were, let's say, 100 nanometers apart. The molecules themselves are only 2 nanometers in size. What I will see, as you know by now, is one single image, one spot. Okay. Now here you might say, okay, how do you know those are single molecules and not two molecules sitting next to each other? What we do is we look for what I call single molecule photobleaching steps. I will show you some examples of those. The idea is if there are two molecules, the molecules themselves are continuously emitting light. If they are photobleached, then they will be giving out no light anymore. And once that happens, the emission will drop down to 0. If there are two molecules, they will drop down to 0, but the chances of both of those molecules, because they are not talking to each other, dropping down to 0 at the same exact time is very, very close to being infinitely small. That we take advantage of to identify whether there were two molecules, so there will be one step photobleaching step, two photobleaching step, or whether there was only one molecule and then it went to a single step to the 0 intensity. So that's how we kind of try to identify those behaviors and recognize the fact that there is a single molecule. If you are interested in kinetics, which is happening very, very fast, let's say something that happens in microseconds, that is going to be a limiting factor, because you have to acquire sufficient number of photons within that microsecond. And that's usually very, very hard, because that's the regime over which all these photophysical effects become very, very prominent. I told you that you have to dilute it. If you have too many molecules floating around in your solution and your protein only binds with micromolar affinity, KD of the system is only micromolar, then your events are going to be very, very minuscule at nanomolar concentrations. So, and as soon as you add micromolar concentrations of your protein, there will be a huge amount of background and you won't be able to study it any further. So there has to be special tricks to be able to do that. I told you that you can do photo switching, but how often do they switch on and how long do they switch off for will define how concentrated a sample you can study. For example, even without switching on your laser, if a lot of molecules spontaneously switch on, which happens thermally as well, and or your concentration is very, very high, and hence always some molecules are on, then you will never be able to do this kind of exercise. And then what happens at these very small number of photons is that you have a lot of noise. This is why you don't see a very nice circular image of a single molecule, even though you know that the PSF is supposed to be a very smooth function. That's because the photons that are emitted, they are falling on the detector probabilistically. And that noise scales as the square root of the number of molecules, and that defines the signal to noise ratio of the system. You will notice that there are other pixels which are bright. That's the background coming from the detector, as well as the system itself, the optical system itself, where there might be either floating molecules or something else, which is causing that extra background that you see. Okay, so these are things that you have to keep in mind when you're trying to do it. But let's say you had a pretty image like the ones that I'm showing you here, then what are the things that you could do? Okay, so here's an example of I want to do co-localization. Okay, now I said that you won't be able to distinguish two molecules if they were very, very close together, right? But imagine that you have already diluted out your sample. So now in the vicinity of this, there's no more single molecules of any kind. They are immobilized, and I am looking for binding to this molecule of other molecules. I know there is only one molecule that it can bind to, because everything else is, there is nothing else in the vicinity of this. This would work. Okay, so what you ask yourself is, I have this Psi3 molecule, I have this Alexa molecule, I can see their spots on your microscope, and then I ask myself, when does this Psi5 molecule or when does this other Psi3 molecule bind to that place at exactly the same time? When this happens, a spot of Psi5 molecule will appear at exactly the same place where the Psi3 and the Alexa 488 was there. But more importantly, because you know what you have tagged your which molecule you have tagged with Psi5 and which molecule you have tagged with Psi3, you can also figure out what is the complex composition of the stoichiometry, right? So here's an example from real data where they're measuring the translation loading initiation. Okay, the ribosome 30S is bound to the RNA. It is tagged with a dye, so you will see that, and you know that there is a 30S already sitting there. This molecule has photo bleached, but that doesn't mean that this complex has necessarily come off, because what we see after a little while is that the initiation factor 2, which is tagged with the Psi5 dye, which is red in color in this plot, comes in. Okay, and so does the tRNA almost at the same time in this particular trace. They also photo bleach. Most of the time they are photo bleaching. They are not really falling apart, because we know that the stability is actually quite high. And after that happens, the 50S RNA is able to load on the system. Okay? So if you look at how often these processes happen, you will be able to tell what is the exact temporal order in which this ribosome is assembled. Okay, and this paper, for example, found that most of the time either the IF2 comes first or it brings in the tRNA with itself. Very rarely the tRNA is able to load by itself, and just by looking at these traces and how often this thing happens. Okay, so you don't need to even model this, even though there's such a complex system. There are 4 components that are talking to each other, yet you are able to interpret this data. Okay, so that's the kind of experiment that you can do. What happens if you have the same protein assembling into a higher order structure? You can take advantage of the fact that, again, as I mentioned, that there is a single molecule that is photobleaching, then you will see a single photobleaching step. If you have 2 molecules that are photobleaching, then you will see 2 photobleaching steps. You won't be able to predict exactly when the photobleaching happens, but you will see most of the time 2 photo-bleaching steps. If you have 4 molecules, then you will see 4 photo-bleaching steps. Take advantage of this, and you just ask, how often does it show this kind of 4 photo-bleaching step versus 2 photo-bleaching step, and that's what the stoichiometry of your system is. Okay, and this kind of approaches have been used where people have been able to figure out the stoichiometry of, let's say, a receptor on the membrane using tags on each of these systems. So here is a system which has a single tag, and you find on the membrane there's a single photo-bleaching step. The NMDA receptor where the subunit is labeled, you see 2 photo-bleaching steps. If the CNG channel has been labeled, then you see 4 photo-bleaching steps on the membrane. Okay, so this is published quite a long time ago, where you can take advantage of photo-bleaching to identify the complex stoichiometry. Okay. The problems that you need to be aware of in this case is that there is going to be insufficient labeling. There's never going to be 100% labeling. So you will be very, very lucky. You have to be if you got 100% labeling, right? The GFP that has been tagged fluorescently, that might not have matured, right? If it is a process that you're looking inside the cytoplasm, there might be, before you start imaging, you have got yourself the bleaching of the molecule has happened, and then you were not able to pick that photo bleaching step, and several other blinking, triplet state. So all this causes problems in counting like this, and you will have to worry about. So let's say your complex forms a system which is a trimer in this case, but because of inefficient labeling, you have got either all of these labeled. So this will show a 3 photo bleaching step, but then there are complexes which will give you 2 photo bleaching steps. A dimer also gives you 2 photo bleaching steps, right? And then you might also have a trimer or a dimer or a monomer give you single photo bleaching step because of this inefficient labeling. So you have to be careful that your labeling has to be high enough. Or what you have to do is you have to be able to estimate what is your effect, amount of inefficient labeling. Because if you do, then you can use mathematical statistical laws to figure out what should be your distribution like. Okay, so this is just to highlight the fact that if you do not have this theta, which is the labeling fraction or detection fraction estimated, then a system which has, let's say, only a complex of 4 can also be fit to a complex which has 5. Okay, just by changing a theta value by a small bit. And this becomes increasingly difficult when the number of complexes that you're interested in has a very large number of proteins in your system. So in that case, the ability to be able to distinguish a 9-mer from a 10-mer is going to be very, very small, because now only few people will worry about this, but I will show you how we have solved some of these problems. The second method that I want to, or at least you will see a lot, is the single molecule tracking method. As soon as people realize that they can pinpoint the centroid of the molecule much better than the diffraction limit, now the molecular scale of motion became accessible. Okay, a protein which is moving at a few nanometers per unit time, like a motor protein, can be studied, even though if you look at the image of that, you might not be able to see much of a difference, right? You won't be able to see a single step. But so then this classic paper from Paul Selvin's lab, what they did was they attached dyes to the myosin 5 motor protein, and they would then let them move on the microtubule. Okay, and as they would move on this, and as they would move on this, you will be able to fit the centroids and look at the movement of the centroids, and you would notice that they are taking steps which can be tabulated, and you can build a histogram of those. And what they found was, of course, that there are 2 kinds of steps. The first set of steps were very, very small, of the order of about 30 nanometers, or sometimes a little larger, where they could not distinguish it, or they were about 70 nanometers or larger. And you will see that these two distributions are slightly different. This is basically the time it took to take that step. Okay. The reason the 70 nanometer steps took longer to happen is because they were two steps which did happen, just that they could not be detected during the imaging that was happening in that time. Yet the time signature, the time distribution, and I can talk about this at a later stage, shows this kind of a Poisson distribution. This is what we call as Poisson distribution, which is characteristic of two steps. One step is hidden, and that's why you have this kind of a distribution for time it takes for you to take this number of steps. Okay, so you can infer that from just looking at that, and by doing this exercise, they could prove that the myosin was taking 37 nanometer steps every time it walked. There's a very similar experiment, but this time around the protein is diffusing on a DNA molecule. So the DNA, if you tag it, it has been extended by applying a flow into the system. And when you take away the DNA stain, you put your labeled protein, these proteins will move around on the DNA. And you can track their motion in time, and you will notice that they are doing what we call the Brownian motion, random motion, going to and fro. What it's doing is looking for a lesion in the DNA, which this protein is known to repair. Okay? And the characteristic of these Brownian motion is that while they will move backward and forward relatively randomly, their net displacement from their original position is going to be mostly centered around the origin. Okay, and in fact, we can predict this distribution, which is given by the diffusion coefficient of that particular molecule. So this is diffusion on the molecule on the DNA strand. You can imagine that this could be a 2D motion, for example, a receptor protein which is trying to find its heterodimer by diffusing in 2D on the lipid bilayer membrane. They can also show different kinds of motions that have been known to happen, and you can get completely Brownian motions, which is completely unrestricted Brownian motion, or get these kinds of fractional Brownian motions, which are limited by things that are obstructing their positions. Or you can get these kinds of super diffusion motions where active process makes it travel larger distances than you would have normally traveled. And you might wonder, how do I distinguish from this random squiggle what kind of a motion I am getting? So at least some of the standard analysis that is done from these single molecule trajectories. So all that has been recovered from these single molecule images is the centroid, and each centroid as a function of time is plotted in this trajectory. If you take these centroids, so let's say this is an example of a centroid plotting that is shown here. You take each of these trajectories and you find out how much is the squared displacement given by this expression that it has moved from its centroid. Sorry, my bad. This is the 2D Gaussian fitting that has been done to this. What you're doing is you're calculating x2 minus x1 squared plus y2 minus y1 squared, adding them together, and that squared distance is what is plotted as a function of the lag time, which is the time from this point to this point, or the next point, or the next point, color coded for how long this distances have been traveled. For a purely Brownian motion, this is going to be, this mean square displacement is going to be linear function of the lag time. That's something that has statistical mechanics and statistical laws, that something that we know of for Brownian motion. If you have a process which is confined or is traveling slower than the Brownian motion because of obstacles in the path, then this will bend towards this. It will travel less distances as it goes, and you can fit them to these functions to extract whether you have a confined motion such that the molecule can't travel further than what it should have been because it's kind of trapped in a particular region, as people do for lipid rafts, or they might be you might be able to fit them to this anomalous diffusion where an alpha exponent on T kind of represents the amount of obstruction that you have, or you can have a model that fits where there is sufficient level of active transport going on that is causing this to move faster than the Brownian motion. So by fitting these kinds of models, you can now extract what kind of a process this is going through. I might say, am I not fitting all the single molecule data? And I will show you some examples where you might not have to fit. There's a lot of things you can extract even from the single trajectories as we go along. Now, what you can do there, what I showed you is that single particle was being tracked, but you saw that to be able to track those single particles, you have to have only a single particle visibility. You might say not all proteins are going to be present in those kinds of densities. How do I track a receptor which is present in very high densities? You can take advantage of the fact that you can again activate a small subset of molecules on your system, and then you can track those particles till they photobleach, just like I showed that E. coli system. And then you might be able to switch on the next set and do this exercise. And that's what has been done many number of times. So this is data where VSBG protein has been tracked on the surface of the cell membrane. And you will notice that some proteins, so this is the diffusion coefficient. So lower values of this represent immobile molecules. High values of these mean high mobility molecules. And you can clearly see that there are lots of proteins which are stuck, not moving around, where there are others which can move around quite a bit. And you can't tell them from their tracks unless you look at individual tracks. But that has been done for you for this exercise. Here's another example of a study that was recently published, where this is ribosomes and the tracks of ribosomes. And what has been done is to identify what is the diffusion coefficient for free ribosomes versus bound ribosomes. Small drug rifampicin has been added so that it stays bound or it's always moving around. And when you do that exercise, you look for the distribution of the ribosomes which are moving fast versus which are moving slow. And you will notice that the free ribosomes, for example, seem to be evenly distributed. So this is the dashed thing is the even distribution of the ribosomes, whereas the ones which are bound, they seem to be populating mostly on the outside, so away from the nucleoid of the bacteria. So you can now start to take advantage of the fact that you have access to mobility of the molecule inside, and hence probably some with the system that allows you to distinguish between what might be happening. So this is a common technique that is used to study what molecules. So I'll give you one more example of this. So here's the same trick. The bacteria has some autofluorescence of its own. You put a freely diffusing protein, this kind of travels everywhere. But when it binds to the DNA, the nucleate of the DNA, nucleate in this bacteria is relatively immobile compared to everything else. So when it binds to the DNA, let's say it's a transcription factor, which is fused with the fluorescent protein, then this gives you a bright spot right at this point. And this is what you saw in your example previously. So in this case, for example, when they were trying to measure the lac repressor, whether it is bound to the DNA or when does it come off, all they had to do was continue to look at the lac repressor bound. This is the state before induction, because the lac repressor stays bound in the absence of the IPTG or the inducer molecule. But when you add the inducer molecules, then everything kind of falls apart and you no longer see that bright spot. Okay. Same goes here. The reverse experiment has been done. And by measuring how much time does it take for these molecules to reappear, you can find out what is the binding time. That's all this that had to be done. Okay, so the time it took for one transcription factor or a certain set of transcription factor in the bacteria to find its target site could be measured just by doing this particular experiment. Of course, you can do more complicated experiments where you can estimate how much time does it spend on the DNA and how much does it move on the DNA. So here's an example of that too. here you have a bacteria. If you image for very long periods of time, let's say expose it for a very long period of time, the protein is doing several kinds of motion. It's moving around in 3D. It is also moving around and once in a while binding to the DNA. And then when it binds to the DNA, it's also kind of diffusing on the DNA for a little while. Okay. So how do I see all of this? You can reduce your exposure time to very, very small time scales. And if you do that kind of a small time scale, you take advantage of this stroboscopic illumination, where you illuminate for a very small period of time, and you can capture very, very fast processes. And if you do this exercise, you now start to see these transient binding events, which are close to this 10 millisecond or smaller. Okay, and by plotting how much these spots spread with compared to a diffraction limited spot, you will be able to estimate how long this thing stays bound. In this case, it is staying bound roughly for about 5 milliseconds or so. Okay, beyond that, it kind of spreads out. So you can take advantage of that, and you can find out how much time it stays and how much does it diffuse on the surface on this bacteria as it's doing this exercise. So by doing this kind of non-specific binding measurements, taking the data from the time it takes to do the search process on the whole bacteria and the time that it takes for it to bind to the DNA, you could extract what kind of motion does it do to find this binding site on the bacteria. And that's what was done in this particular protein, this particular paper, sorry. Okay, so you saw epifluorescence, high-low. Here's a new method that was developed a few years ago, where they took advantage of light sheet microscopy to now do selective illumination in a small volume. And this was needed because bacteria is 1 micron. So very easy to do single molecule imaging. You don't need to worry about the background coming from the rest of the volume of the cell. Mammalian cells, of course, as soon as you get to that stage, will have a lot more volume. The epifluorescence, if you switch on your laser at the same intensity required for imaging a single GFP, you will already see the cell is bright, even if it doesn't have any bacteria, any GFP proteins. And you cannot use the regular light sheet microscopes. So what people in this did was they would use this reflected light sheet. So this is acting as a small mirror for the cell. This is usually an AFM tip that has been coated to act as a mirror. The laser comes from the top and then is reflected into the cell. And then the laser is also squeezed in one axis, so that while it focuses on this axis, along the other axis, it is a flat sheet. I will show you a kind of a scheme for that and a video of that. So this is the mirror that I'm talking about. The cell is sitting here, and the light sheet is now coming top down on hitting this area, and then it's going to get reflected into this, creating an optical section through the cell, mammalian cell in this case. So this is, let's see if this plays. So this is a view of what is being done, okay? And by scanning the cell, instead of scanning the whole geometry here, you can get Z-sectioning across the cell. I might say, why did they go through all this trouble? Why not do a confocal imaging? You will notice that the whole of the cell is illuminated in this case at the same time. Also, you will have an objective, which is a high NA objective that can capture all of this imaging at the same time onto a detector. This is how the setup looks like, but I won't spend time describing it for the lack of time. What I wanted to see is this. So this is high-low, the technique where you create a very thin sheet, still has a lot of background, but when you create a reflected light sheet, you can get very high contrast and you can see single transcription factor. So this is basically a nuclei, an odd-shaped nuclei, but it's still a nuclei, and you can see transcription factors which are bound to the nucleus. If they were freely floating around, then you would see a blur. The only reason you see them as a spot is because they are transiently binding to the and DNA, and that DNA is still mobile, but it's not as mobile as which will kind of diffuse out the signal. Okay. All right. I do not want to take you through all of this. These are similar techniques. What you could discover, at least doing this kind of an exercise, was that each of these transcription factors, so this is estrogen receptor, this is glucocorticoid receptor, how they were binding to this bound DNA and how long they were staying there. And from there, you can infer the mechanisms by which they operate and find their sites, but also the mechanisms by which they would interact with the DNA and how long they will stay on the DNA. Okay.